Hello and welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. I'm Sassan Ostvar, and together with Sarah Light, it is our pleasure to moderate today's session, highlighting the outstanding work of our postdoctoral members, Leah Greenspan and Ben Cox. Each presentation will be 20 minutes long, followed by Q&A. And we do want to invite you to use the Zoom Q&A interface to enter your questions for the speakers. To begin, it is my pleasure to introduce our first speaker, Dr. Leah Greenspan. Leah graduated magna cum laude with a distinction in research from Cornell University, where she majored in biological sciences with a concentration in genetics. Under the mentorship of Andrew Clark, she researched the variation in X chromosome genes and their association with male reproductive success in Drosophila. She then joined the lab of Jennifer Zallen at Memorial Sloan Kettering as a research technician, where she worked on genetic screens to discover new genes that disrupted axis elongation in Drosophila. She received her PhD from Johns Hopkins University School of Medicine's Biochemistry, Cellular, and Molecular Biology program in the lab of Erica Matunis. Her thesis work focused on the regulation of niche plasticity in the Drosophila testes, and she received a Ruth L. Kirstein NRSA Individual Predoctoral Fellowship for this work. Leah is currently a postdoctoral training fellow in the lab of Brent Weinstein in the Division of Developmental Biology at NIH, where she has established, among other things, a cutaneous wound healing model in adult zebrafish to characterize the changes in wound healing during aging and diabetes. Leah's work has been widely recognized through various awards, including an NICHD Intramural Research Fellowship, NIH Fair Award, NICHD Early Career Award, Development's Pathway to Independence Program Fellowship, and K99 NIH Pathway to Independence Award. Today, she will be sharing her work on cutaneous wound healing in zebrafish. Leah, please take it away.
Well, thank you so much for that very nice introduction, and thank you to the organizers for this really amazing opportunity to share my work with you all today. As Sassan said, I'm Leah Greenspan. I'm a postdoc in Brant Weinstein's lab at the NIH, and today I'm going to discuss my work on the investigation of cutaneous wound healing in zebrafish. So we all have gotten scrapes and cuts throughout our lives, and for most of us, we're able to heal those wounds quite quickly in about 4 weeks' time. And this is what's known as acute wound healing. But for about 2% of Americans, they actually suffer from a delay in wound healing or wounds that never fully heal. And this is known as chronic wound healing. And this is particularly prevalent in the elderly population, as well as those that suffer from diabetes. And you can imagine if you have wounds that fail to heal, they're very susceptible to infection. This can lead to necrosis and even amputation in some cases. And it's thought these chronic healing conditions are due to a delay in one or more of the different stages of wound healing. So wound healing occurs in a very stereotypical fashion, where you first have your hemostasis phase, in which a blood clot is going to form, followed by the inflammation stage, where you're going to get the recruitment of many different immune cell types to that wound site. That will then lead to the proliferation stage, where you're going to get the proliferation and regrowth of tissue into that wound site, followed by the remodeling stage, where blood vessels will get pruned and collagen will get remodeled. Now, many studies on wound healing have been done in different mammalian models, but it's been really challenging to understand the cell-cell interactions that are occurring because it's been hard to live image these wounds. So I wanted to use a model that's more amenable for live imaging. And so I wanted to use the zebrafish. So zebrafish are known to be a really great model to study developmental processes live, but we also have adult zebrafish that we can image. So here I'm showing an example of a zebrafish that's pigmentless, so it has mutations in the pigment cell, so it's transparent. We also have access to many different transgenic lines that label all different cell types. In addition, we've developed intubation techniques. As you can see here, this fish getting intubated and has anesthesia water running through it. And this allows us to do more long-term time-lapse imaging for up to 24 hours at a time. In addition, mammals and zebrafish actually have a very similar skin composition. So both of them have a stratified epidermis layer that's on top, that's then followed by the dermal layer that has lots of fibroblasts, and then the hypodermis layer, which consists of many fat cells on top of that muscle layer. That then leaves the question, well, how can we actually injure these fish to start to study wound healing in them? And so during my postdocs, I've developed an injury method using a rotary tool to create a cutaneous wound. So here you can see the rotary tool on its stand. And then if we zoom in, we can see that at the tip of the rotary tool, we have a flat end drill bit, which has 4 very sharp blades, but a flat end. In addition, we use CAD design and 3D printing to produce a cowling, and that helps me actually control the depth of the injury so it doesn't go too deep into the fish, as well as this holding platform, which allows me to anchor the fish so that it stays taut while I'm creating the wound. So this is what it looks like with a anesthetized fish on that holding platform anchored in. You can see the cowling has come down and is creating an injury on the flank of the fish, and you hold it for about 10 seconds to generate that wound. And so this is what the wound looks like, where you can sit again on the side of the fish. We can create that cutaneous wound. And so I wanted to better characterize this injury model and look at different histology sections through the wounds. So here I'm showing you a fish before injury. And if we do a transverse cross section through the fish, again, what you can see is you see this epidermis layer of skin on top of the scales of the fish. And then underneath those scales are the dermal cells. Then your fat cells make up the hypodermis on top of your muscle. But after injury, again, we can see the nice cutaneous wound on the side of that fish. And then upon injury, we see all of those layers of skin have actually been injured up into that muscle layer. And so this is what's known as a full thickness wound when you injure all the layers of skin. Then if we look 15 days post-injury, you can see that most of that wound has recovered. And if we look at the different cell types, we can see that all the cell types have returned and repaired that injury. So it seems that it takes about 2 weeks and then another week to kind of thin out those layers back to pre-injury conditions for these wounds to heal. So what I really wanted to start to understand is how do we go from this injured state to this repaired state? And can we use live imaging to start to understand the cellular behaviors that are driving this repair? In particular, I wanted to know if we can use zebrafish to study the reepithelialization of the skin, the inflammation stage and the recruitment of different immune cells, and how blood and lymphatic vessels regrow during the proliferation stage, as well as their remodeling. So first, I wanted to look at the reepithelialization. So here I'm showing you a fish whose beta-actin is labeled in GFP, which labels all the cells, but it also nicely labels the skin over the scales. And so this is the flank of the fish, and we can kind of zoom in on a section where that injury is going to occur. You can see it nicely intact. And then upon injury, you see that the skin is no longer in that injury site. But what's interesting is that we immediately start to see skin from the surrounding area of the wound streaming into that wound site, as well as other cells within that wound, but that wound is sealed up very quickly. And here we're showing it 2 days post-injury. So I then wanted to look at this a little bit more carefully, and I got a keratinocyte specific line, which labels the epidermal cells of the skin. And here you can see that before injury, you can see a nice top-down view of what that looks like. And this is a side view. You can see that really thin epidermis layer of skin. Upon injury, again, you see that there's no skin anymore in that injury site. But by 3 hours post-injury, we see a significant closure of that wound site. It's almost fully closed at 6 hours post-injury. And then by 1 day post-injury, we see complete closure of that wound site. So the skin has re-epithelialized over that wound. So in zebrafish, we see that this re-epithelialization phase is very immediate and is very quick occurring. And you can also see kind of the depth of the injury here in this side view. So in addition to looking at reepithelialization, I really wanted to start understanding this inflammation stage, as it's been shown that in chronic healing conditions, inflammation can be aberrant and can either be delayed or it can be prolonged that affects wound healing. So here I'm showing you a fish in which the skin has been labeled with a Boddippy dye in cyan, and the neutrophils are labeled in magenta. And you can see that the neutrophils are actually kind of scattered throughout the skin before injury. But upon injury, we start to see these neutrophils being actively recruited to that wound site. So you start to see a lot of migration of neutrophils nearby, as well as those further away being recruited into that wound. And so I wanted to kind of understand neutrophil behavior more in depth. And so I did more high resolution imaging where I could actually track individual neutrophils. And so what I saw was this initial migration of neutrophils towards that wound site, followed by this amplified recruitment where you're getting neutrophils from further away. And we see that occurs in an exponential fashion over the first 16 hours post-injury. In addition, we start to see these neutrophils clustering together, and we can quantify that by looking at how a single neutrophil, how close it is to its 9 nearest neighbors, and see that that distance gets closer and closer together over time. In addition, we start to see these neutrophils infiltrating that wound and going deeper down into that wound site. And we can quantify their speed. So if we look at neutrophils in an uninjured fish compared to an injured fish, we see a significant increase in the speed of these neutrophils. And so all these behaviors are what's known as neutrophil swarming and has only been reported to occur in small dermal wounds that they make in the ear of a mammal that they can image for about an hour or two. But because we can image for much longer, we can look at these behaviors and quantify them in these bigger cutaneous wounds. So in addition to looking at how neutrophils are recruited to the wound site, I wanted to know, well, how long are they actually staying within the wound? In particular, here I'm showing you before injury where, again, you can see the neutrophils scattered, and by 2 hours, we start to see them along the periphery of the wound. But by 1 day, we see that wound is completely covered with neutrophils. But interestingly, 2 days post-injury, we start to see a lot of neutrophils that have left that wound site, and they continue to leave over a few weeks' time. And so if we quantify that, what we see is that there's this significant increase in neutrophil recruitment at 1 day post-injury that again goes down by 2 days. So it seems that neutrophils respond immediately to injury, but are only in the wound site transiently to help repair and get rid of debris. So in addition to looking at neutrophils, I was really interested in understanding how the macrophages were behaving, as it's been shown previously that macrophages play a dual role in wound healing, where they have this pro-inflammatory role as well as this pro-reparative role that they can play. So here I'm showing you a movie where you can clearly see before injury, the macrophages are all over the skin. But upon injury, you can start to see the macrophages moving into that wound site. But unlike the neutrophils that were actively migrating into the wound, the macrophages seem to kind of be moving into that wound by kind of hitching a ride on the skin as the skin streams into that wound site. So it's a bit different migration pattern that they're playing as they move into that wound. And so I then wanted to better understand, well, how long does it take for them to get to their peak recruitment? So here I'm just showing you what it looks like before injury. After injury, you don't see many macrophages immediately in that wound site, but start to see some at 1 day. And we can continue to see them being recruited to the site of injury and go out to several days post-injury. And so what we quantify this, what we see is that macrophage recruitment actually peaks at 10 days post-injury compared to the neutrophils, which are peaking at 1 day. And so this dotted green line is just showing the amount of macrophages normally within that area. And we can see that there's a significant increase in number at 10 days, and it seems to be elevated even out to 30 days post-injury. And so it's really interesting that they're taking a lot longer to be recruited. But what is going on with their activity? What kind of role are they playing in that wound site? So in order to look at this, I wanted to get a sense of how activated the macrophages was. And it's been shown that you can assess this by the shape of the macrophage. So a macrophage that is more protrusive is actually going to be surveilling and in a less activated state. While as it gets activated, it starts to bring in those protrusions and round up and become more activated. So we can actually quantify a roundness factor for each macrophage to understand the distribution of these cells within the wound. So if we look at an uninjured wound, what we see is that there's about 50% coverage of less activated macrophages in that wound site, and about 17% are in that activated state. But upon injury, we actually see the significant increase in the amount that are in that more activated state. And this starts to decline at 7 days and then continues to go back to a normal distribution by about 10 days. And so the way that I've kind of interpreted this is looking at how they're most activated in between that 1 and 7 days during that pro-inflammatory stage. And then they kind of switch back at 10 days post-injury where they're still getting recruited. And that seems to kind of be the switch to that pro-reparative phase. And so I'm interested in kind of looking more into the macrophages and understanding what the signals are and how that might correlate with these different stages that they're in. So in addition to looking at the inflammation stage, I was really interested in looking at how blood and lymphatic vessels regrow into that wound site. As you can imagine, it's really important to have blood vessels reestablished, blood flow back into that wound site to allow oxygen and nutrients to help with tissue repair. In addition, lymphatic vessels are really important to help drain any excess fluid that's occurred. And so here I'm showing you an adult zebrafish with the lymphatic vessels labeled in green and the blood vessels in magenta. And you can see these lymphatic vessels make this beautiful patterning where you have this lateral lymphatic vessel that runs along the length of the fish. And then if we zoom in, we can see these superficial intersegmental lymphatics that run perpendicular off of that lateral lymphatic. Then if we zoom in further, we can see what the blood vessels look like, where you have these skin blood vessels on the surface that are more tortuous, and then these really thin underlying muscle blood vessels that run along the somi. So before injury on the flank of the fish, you can nicely see the patterning that I just described. But upon injury, you can see that that wound area is devoid of both types of vessels. But by 2 days post-injury, we start to see sprouting from the vessels that are surrounding the wound. And then we see a lot of growth at 4 days post-injury towards the center of that wound site. And then a continued growth until about 10 days where all those vessels have kind of fully covered that wound site. And so we can quantify this rate of growth. And what we see is that this occurs in 2 phases, a fast phase over the first 4 days in which the vessels are growing, and then it tends to slow down and plateau over those next several days as they continue to grow into that wound site. And so I then wanted to look at, well, how are the vessels kind of patterning during that regrowth stage? And so to look at that, I did more high resolution imaging looking specifically here at lymphatic vessels. And so this is showing all lymphatic vessels with a single lymphatic vessel pseudo-colored in green. And you can see that this lymphatic vessel runs from this lateral lymphatic vessel that's on the midline of the fish all the way down to the bottom of the fish. Now, upon injury, what happens is you can see that this vessel gets severed. Then by 2 days, we start to see some sprouting. We see a lot of growth at 4 days, some branching occurring, and then we see a disconnection, a reconnection, and this continuous remodeling occurring of this lymphatic vessel. And I've taken the same fish out for 2 months post-injury. And so this has been shown to occur in humans too, that vessels can actually remodel for several months or years after an injury. And we're seeing that happen as well in zebrafish. And so I'm really interested in now understanding, well, do these vessels ever go back to the patterning that they were before an injury? And if they don't, are they just as functional as they previously were? Or does somehow regrowth after an injury kind of affect the function of these vessels? And so these are some of the questions I'm interested in studying in the future. And so I hope today, so far, I've convinced you that zebrafish are an excellent model to study cutaneous wound healing combined with live imaging, that we can really start to look at the cell behaviors during the different processes, such as the reepithelialization stage when the skin regrows, the inflammation stage when you get the recruitment of different cell types, the proliferation stage of blood and lymphatic vessels regrowing, as well as the remodeling stages as lymphatic vessels are pruning back. And so by taking live imaging of all these different stages, I'm able to develop a timeline of what acute wound healing looks like in zebrafish, where you have this really fast reepithelialization that occurs immediately, followed by an inflammation stage that lasts about a week, proliferation that can go on for about 2 to 8 days, and then remodeling that can go on for much, much longer. And so now that I've established this timeline or this baseline of what's supposed to occur during acute wound healing, I'm now really interested in understanding, well, what's going wrong in these chronic healing conditions? Are cells behaving differently? And can we start to understand if that's similar in different chronic conditions, or is it always or is it different things that are going awry? And so the first chronic condition I wanted to look at was aging. And so here I'm just showing you a young fish that right after injury, and then you can see 2 days post-injury where their wound's already starting to be cleaned up. But if you look at an old fish, a fish that's 2.5 years or more, you start to see that this wound actually still looks quite inflamed immediately and still red 2 days post-injury. And so I've started to do some live imaging on immune cells, the neutrophils, and what I saw is that while in the young fish, these neutrophils get accumulated or recruited to that wound site in this exponential fashion over 16 hours. In these older fish, we actually see a faster recruitment of these neutrophils and a lot more being recruited to that wound site. And these neutrophils are also able to cluster together much sooner, probably because there's way more numbers. And so what I think this might be indicating is that these older fish most likely have neutrophils that aren't functioning as well and therefore require a lot more neutrophils in order to clean up this injury. And so this is what I'm hoping to explore more in the future and in my own lab. In addition, I also want to look at diabetic conditions to see, since it's known that people who suffer from diabetes actually can have a really hard time. They get foot ulcers, ulcers that don't heal. And so I really want to see, well, how are these wounds healing and what's going wrong? And so to do that in zebrafish, I have a collaborator, Mike Parsons' lab at UC Irvine, that has created these fish in which you have an insulin promoter that's driving an M-cherry fluorophore, as well as this nitroreductase enzyme. And so what that does is you can see expression in the pancreatic beta cells that are expressing insulin. But if we add this prodrug metronidazole to the water, any cell that's expressing nitroreductase will undergo apoptosis. So we can ablate these insulin-producing cells so the fish can no longer produce insulin and they become diabetic. And so I have these fish and I'm hoping to do my injuries on them to start to understand changes in cell behaviors under these conditions. And so in addition to looking at these chronic healing conditions, I'm also really interested in starting to understand the molecular characterization of different cell types throughout the healing process. And so in order to do this, I'm planning to do single-cell RNA-seq within these cutaneous wounds at different time points throughout the healing process. In addition to single-cell RNA-seq, I'm also using a ribotag trap RNA-seq technique to specifically look at endothelial cells. We've generated transgenic fish in my lab that have ribosomes that have an HA tag that are expressed in the endothelial cells, and then we can do immunoprecipitation using an anti-HA antibody to pull down on these ribosomes and get actively translated RNA. And so if you're interested in this technique, we have a paper up on bioRxiv that's describing it. And I'm hoping to use this to start to understand the changes, the vascular changes that are occurring throughout the healing process. And so with that, I would just like to thank the entire Weinstein lab, my mentor, Brant, who's been an excellent mentor, as well as all the people in the lab, as you can see here. I'd really like to thank my mentees who helped with all this work, my funding sources. If you're interested in hearing more about this cutaneous wounding model, we have a paper out. If you want to hear more about this endothelial ribotag technique, feel free to check that out. I am currently on the job market, so if your department is hiring and you think I'd be a good fit, please let me know, and I'm happy to take any questions. Thank you so much.
Thank you very much, Leah. Absolutely beautiful. Thank you.
Thanks.
So I'd like to start the Q&A with a simple question for myself. So I was wondering, would you expect any of the rates, especially the reepithelialization rate, to vary depending on where the wound is in the body?
That's really interesting. So I haven't tried different areas yet in the body because of different reasons, but it is possible that that rate could be different. It is quite fast in zebrafish compared to humans. So in humans, that reepithelialization does occur later during that proliferation phase, but I don't think it would differ too much. I think it's a mechanism that fish have adapted in this aquatic environment to help seal up the wound, but there could be changes depending on where the location is.
Thank you so much. And we have a question from Ben. He asks, do you manually pluck scales before the injury? And how deep is the wound you're making compared to a scale pluck?
So I do not pluck scales before the injury. The rotary tool will actually get rid of the scales, the way that it kind of creates the injury. The scales get removed during that injury process. But scale removal is considered kind of a partial thickness wound because you're just getting rid of the epidermis layer and a little bit of that dermis layer versus using my injury system. It's a full thickness wound where you're getting rid of the epidermis, the dermis, and the hypodermis. So you're doing more layers of skin. And so it's a bit deeper than if you were to just do a scale pluck. And therefore, we can look at some differences in wound healing doing this deeper wound.
Okay, and we have another one from Eric Peterman, who says, hi, Leah, super cool talk. Neutrophil swarming and clustering after wounding is dependent on cell-cell calcium signaling. Do you suspect the older neutrophils have some sort of cell-cell communication defect?
That's a really awesome question. I'm definitely interested in starting to kind of look at, is it something with signaling between the neutrophils? Is it signaling coming from the dying cells that's causing the differences in recruitment of the neutrophils? I know we are working on making a GCamp fish in the lab. We have a new grad student, so that could be something that I look at in the future and look at calcium signaling and see if it's affected. That would be really interesting.
Thank you. We have one from Marcia who asks, how long do you intubate the fish in your experiments?
Nice. So for most of my movies, they're about 17 hours long. So that's how long the fish is intubated for. And then we're able to revive the fish after the movies. But I think people in the lab have gone up to 24 hours using intubation. And you can revive the fish and then you can re-intubate them on a later date if you then want to do another long time lapse. So it does work pretty well. I mean, it's not always perfect, obviously, but it's been really a great technique for doing some of this long-term live imaging.
And Robert Cornell asks, hi Leah, for the multi-day time lapse, I assume you had to remount the fish every day. Was it very challenging to orient the fish the same way so precisely?
Thanks for the question. So for the time-lapse imaging, I usually just take one image every day or once a week or every couple of days. And I always make sure that my fish the head is on one side and the wound is down because it's an inverse microscope. And I am usually able to really find my wound. As it's healing, it does get more difficult finding the wound. So I do have to use other parts of the fish to orient myself to make sure I'm getting the same area every time as things are healing. Usually you can kind of look at the scale patterning because the scales take a bit longer to come back. And that can help orient you where the wound was to make sure you're getting the same site every time. But they don't get mounted in agarose as adults. We just mount them in a dish with some water and put a sponge on top to keep them down while we're imaging. So it's not too difficult to mount them.
Okay, fantastic. Thank you so much again, Leah. Beautiful talk.
Thank you.
I'd like to remind you that you can keep asking questions in the Q&A. And with that, we're going to go to Sarah Light for the next speaker. Thank you.
All right, thank you, Sassan and Leah. So my name is Sarah Light, and I have the pleasure of introducing our second speaker, Dr. Ben Cox. Ben received his bachelor's in biology from the University of Texas at Austin, and from there he went to Duke University for his PhD in the Developmental and Stem Cell Biology Training Program. For his doctoral work, he was mentored by Stefano D'Italia and Ken Poss, and worked on investigating osteoblast and epidermal cell movements in regenerating zebrafish scales. And for the last 5 years, he's been working in the lab of Selena Giuliano at the University of California, Davis on ECM remodeling and progenitor cell migration and invasion using a Hydra model system. He's received many awards over the years, including the Hartwell Foundation Biomedical Research Fellowship and the Emerging Organism Research Grant from the Society for Developmental Biology. And a fun fact I found in his CV, while a grad student at Duke, he hosted a radio show covering science and research policy news called The Endless Frontier. And I'm very excited for him to share his work on ECM remodeling with us. So without any further ado, Ben, the stage is yours.
Thank you so much for that lovely introduction, Sarah. Okay, am I good? Yeah, so thanks so much to SDB for the honor of selecting me and giving this talk. I really enjoyed Leah's talk as well. I did somewhat similar work as a grad student, and just seeing that fish being intubated, just like a flood of memories of very, very long nights in the lab came back to me. So really cool to have her work, to share a seminar, an hour with her work. Getting into what I'm doing now, though, I am a postdoc in Selena Giuliano's lab at UC Davis, and my work relates to remodeling of the extracellular matrix during heterogeneration in Hydra vulgaris, and how that helps get stem cells get where they need to go. Just to orient you, the Hydra vulgaris is a small freshwater cnidarian capable of whole body regeneration. It's the primary species we study in the Giuliano lab, although I'll talk about maybe some other ones at the very end of this talk. So it has a pretty simple anatomy where a body column, which is really just kind of a hollow radial tube, separates a hypostome containing the mouth and tentacles from the foot, which is used to attach to substrates. And you can take a very small piece of tissue, as small as a couple hundred cells, and cut it out and get regeneration of an entirely new animal within a couple of days. You'll see this timestamp up here measures hours and minutes. So within just under 2 days of this movie, you see the reformation of a mouth and tentacles. So any animal that's capable of such astonishing feats of regeneration should have a lot to tell us about the cell biology of tissue regeneration. Although before I go into that, I think I would be remiss if I did not mention the namesake of this seminar series, Ethel Browne Harvey, who just discovered the organizer in Hydra viridissima. Actually, in her lifetime, she was much better known for her work on sea urchins, but it was her work as a grad student on Hydra viridissima where she did some really pioneering work on organizers and axis induction. So in 1909, she transplanted head tissue from one green hydra called Hydra viridissima onto the body column of another and induced new body axis formation. This, of course, was quite similar to the work that would later be done by Hilda Mangold and Hans Spemann, resulting in a Nobel Prize for Spemann. This was in 1924, 15 years later. So Mangold was not honored with the Nobel Prize because she passed by then, and they don't award it posthumously. But of course, the significance of Browne Harvey's work was not really appreciated at the time. And there's a nice paper from Howard Linhoff about 30 years ago looking at this as a sort of a case study and saying it raises the question about possible interplay of basic prejudices, those of medical scientists in general and the Nobel Committee in particular, in using anthropocentric criteria, and then just of male scientists in evaluating the work of women without significant academic rank. So I'm glad that we're honoring the work of Dr. Browne Harvey, even if somewhat belatedly. So anyway, just to get back to the science, my main question is, what is the role of extracellular matrix remodeling in regeneration? And this is going to kind of come in two parts. The first of which being to what extent the ECM is remodeling and how that happens. And then what is the broader role of ECM remodeling in regeneration, specifically the renewal of the lost cell populations, because traumatic injury to the hydra does cause the loss of so many cell types. So one of the things that I hope to convince you all of and convince potential hiring committees in the future of is that the hydra is a really nice model for studying the role of the extracellular matrix in homeostasis and regeneration, because it has this really simple body plan that kind of just gives you the bare bones to study the extracellular matrix. You have these two epithelial layers, endoderm represented in magenta, ectoderm represented in yellow, and they are separated by this basement membrane-like ECM that in the field we sometimes call the mesoglia. I'll just call it ECM here. But the main components of hydra ECM, which you can see in this overlay of fluorescence on bright field, are laminin and collagen 1. And collagen 1 is really kind of sandwiched in between laminin as well as a collagen 4, although that's not shown here. But the dynamics for these proteins are broadly similar during regeneration, so I will focus on collagen during this talk. But the other nice thing about hydra is that their ECM is composed of these very well-conserved proteins. So fibrillar collagens and laminins, which I showed before, as well as matrix metalloproteases and other ECM proteases, arose at the dawn of Metazoa. As cnidarians split off from bilaterians, including all the lab animals we know and love, like fish, mice, and flies, they expanded these gene families, evolved new genes like fibronectin and tenacin, but cnidarians maintained these core components, and so we can use them as a sort of comparison for structures, parts of the ECM that have evolved and parts that have been conserved over hundreds of millions of years. So before I get into showing you hydra head regeneration, I just wanted to return to this basic time course, really showing you in still images what was in that video, because especially early on, it's not going to look like much. There's just a little stump, essentially. And that's what a lot of the images I'm going to show look like. So I will always orient you by showing the head at the top, foot at the bottom, labeling structures like regenerating tentacles. So yeah, this is what we'll be seeing over the next handful of slides and minutes. So one of the first things I wanted to do was just look at the expression of collagen during head regeneration. And one of the a strange thing I found that had briefly, but not really in much depth, been described before, was that immediately after closing the wound, you actually see a significant gap in the expression of ECM proteins at the head wound. So if we overlay with actin phalloidin, you can see that these are indeed this is indeed a closed wound, a cellularized region. It's just that collagen is not making it all the way to the end of the wound. And this gap in ECM expression actually is maintained for several days, and then eventually gets filled in by synthesis of new ECM proteins, in this case collagen. And we can quantify that using just a relative fluorescence in the head wound region compared to elsewhere in the same animal's body column. You see that is steadily filled in by new collagen synthesis. So my main question is, how is this remodeling happening? You have a wound, and then an immediate wound closure, but somehow after that wound closure, you actually see a decrease in the amount of collagen. So is this actively being broken down, and how? And then after it is broken down, how is it built back up? So to answer this question, I looked at the role of matrix metalloproteases. I briefly mentioned those on my ECM evolution slide, but these are, as the name suggests, a family of proteases that has a metal ion cofactor and is broadly active in the degradation of extracellular matrix proteins. So what I found was early in regeneration, at one day, you do have this relatively large gap in the ECM, but when you treat with the broad spectrum MMP inhibitor merimostat, you see a significant decrease in the ability of this collagen gap area to form. Not only that, but if you continue treating for several days, you'll see a significant reduction in the formation of new head structures. So significantly fewer tentacles formed by the animals. So going from this broad spectrum inhibitor, I wanted to look at what specific genes might be involved in this breakdown. So I looked at using all of the transcriptomic data we have and identity of matrix metalloproteases. I identified a handful that are expressed during head regeneration. I'll just show one since they in situs look somewhat similar. You have this gene MMP14 that in the uninjured animal is expressed in the foot and in the tentacles, although not really in the head proper. But then when you look early in regeneration, this closed wound where the head is going to start regenerating, you see this really nice dark stain. And we can confirm through previously published bulk RNA-seq data that we see a significant increase in head expression. Although this does not, because this is bulk RNA-seq, we're not looking at cell identity, but we can see if we just look at somewhat higher resolution that these are indeed expressed, these MMPs are being expressed primarily in the endoderm. So it's endodermal tissue that seems to be creating the enzymes to break down extracellular matrix. Further, so we know that we need to break down ECM, but what role is new collagen synthesis playing in head regeneration? So similarly, I turned early on to some pharmacological treatments using a drug called dipyridil that inhibits the cross-linking of collagen necessary to create their three-dimensional helix, three helix structure. Early on, you see this normal gap formation, but when treating with dipyridil, you completely lose this early synthesis of new collagen protein that you're seeing even as early as 24 hours post-amputation. Also, you're just losing new collagen synthesis in general. You kind of get this animal that balloons up, presumably due to the lack of sort of any elastic structural support provided by the extracellular matrix. And we can see this significant decrease in collagen intensity. Not only that, but if we keep the animal on the drug for a couple of days, we see a total loss of new head structure formations. We have dipyridil here in two two sets of treatments. If I then release dipyridal from one of those sets and keep it on in the other, we can compare the ability of the animal to cover from this collagen synthesis inhibition. And we see, compared to an ethanol control, that when you release the animal from treatment after a couple of days, you do start to see the head regenerating somewhat normally, whereas there's no progress whatsoever under 4 days of continuous collagen inhibition. And we can see here this dipyridal 2-day treatment that then gets washed out to allow for recovery. We see significant recovery of regenerative capacity, although not quite to control levels. So just as with the MMPs, I wanted to know where new collagen synthesis was occurring. And as this is especially interesting because I think there's some old papers that our own data conflict with a little bit. So I wanted to be quite thorough in showing that the new collagen appears to be being synthesized. New collagen transcripts appear to be being synthesized by endodermal tissues. So this is a plot from our single cell database, where here you can see that this collagen 1 transcript is expressed overwhelmingly in the endoderm, especially at the extremities, the head and the foot. And our collagen 1 fluorescence in situ hybridization data agrees with that quite well. We can look in higher resolution here and see, unfortunately, within Hydra, we have an issue with probes really like to stick to the outermost part of the animal. I think that's mostly what's being represented here, as this green does not really appear to be within cells, whereas in the endoderm, it's quite bright. And we can see if you just look at Hooke's nuclear staining, there's a little gap here that represents the mesoglia and the border between endoderm and ectoderm. And so endoderm is overwhelmingly expressing the new collagen, and we can also see that occurring during head regeneration. And of course, this aligns quite nicely again with a previously published bulk RNA-seq data. So just to look as a sort of conclusion of where we are so far, ECM is removed from the site of the head wound in the first day post-injury after wound closure. And MMPs expressed during head regeneration are required for that ECM gap formation. New collagen also originates in the endoderm. So you actually have the same cell layers, the same cells, expressing genes involved in breaking down ECM as well as building it back up. So obviously there's some pretty precise coordination at the post-transcriptional and even post-translational level, coordinating this, choreographing it perfectly. And that's an area of inquiry I'm in the sort of the early stages of looking at as well. So the second part of my sort of two-pronged question about the overall role of ECM in regeneration is, how is this remodeling affecting stem cell behavior? So I showed you this image of the 2 layers of the hydra earlier, and what I neglected to show was that within these 2 epithelial layers, there's actually a third cell layer, the interstitial cell lineage. Their stem cells are located exclusively in the ectoderm, but their derivatives are located in both cell layers, both the ectoderm and the endoderm. And these include cell types like neurons and gland cells, as well as not shown here, germ cells. So my hypothesis really was, maybe this ECM remodeling is playing a role in cell invasion. So cell invasion is the process through which cells travel through the ECM to reach a different tissue layer. And my hypothesis then was that maybe this lack of a full ECM complement is allowing for facilitating faster and more thorough invasion of these interstitial stem cells into the head endoderm, because they've lost so many gland cells and neurons that need to be replaced. So to orient you, I'm going to be working with this nanos GFP line, expressing cytoplasmic GFP in the interstitial stem cell lineage. Many of you probably know nanos from its role in germ cells. And like I said, it does mark germ cell progenitors as well as the progenitors of neurons, gland cells, and cnidocytes. And so you can see that these cells cover the length of the body column, but are more or less excluded from the head and the tentacles as well as the foot. And when we look at cryosections of the animal, we can see these nanos GFP cells are broadly excluded from the head, as I mentioned. And you can see just a couple here down at the bottom of this image, whereas there are tons in the body column. And again, they're only in the ectoderm, excluded from the endoderm. So in the uninjured animal, while they're excluded from the head, but when then you injure, you obviously have tons in the head. And my question was, are these nanos GFP cells actively migrating to the wound site, or are they simply already there by nature of axis relocation when you amputate in the mid-body column? And so my prediction was, because of this massive loss of cell populations in the head endoderm, you would actually need interstitial cells to migrate to the head, as well as invade in sites of low ECM expression. So to answer the first of these questions, I turned to one of our favorite experiments in classical developmental biology, which is tissue grafting. So I looked at a nanos GFP animal, took its bottom half, grafted it to the top half of a non-transgenic animal, got this chimera where I could injure and leave some non-transgenic tissue over which these nanos GFP cells could migrate, and then assayed migration. So if you graft these animals and don't injure, you do still see some migration, but you see quite a bit more when the animal is injured and you look a day later. So you see a significant increase in the migration of these cells, more than just 100 or 200 microns past the graft site. So interstitial cells are migrating to the site of head regeneration after injury. Then the follow-up question is, are they actually also invading the endoderm? So if we look at this nanos GFP line again, with tissue cryosections, you can see early on there are some nanos GFP positive cells in the head wound, but they appear to be mostly on the outermost layer in this ectodermal layer of tissue. While it's a little bit hard to see without the collagen as a guide, they're very far on the outside of these cells. However, if we look a day later, at 1 day total, 24 HPA, we can see that these nanos GFP cells are indeed appearing within the head endoderm where they normally are not present. And here I've just marked where that collagen should be when it's the animal's fully healthy, but we have a much sort of reduced collagen at this early stage of regeneration. You can look at this in much greater detail, see that a couple of these nanos GFP positive cells are sort of just made their way past the border of low collagen expression. And if we can quantify
1 minute remaining.
Okay, we can quantify this and see a massive increase in the expression of endodermal nanos GFP cells. And this is a pretty early experiment that I'm looking to repeat, but we also see this lack of invasion when we inhibit MMP activity. Finally, one thing I'm really excited about looking at is using this detergent called inguinal sarcosine to decellularize the animal and just look at the ECM itself, where you can image it quite nicely without any cells getting in the way of your light path. You can see this really nice increase in the amount of pores in the regenerating head that presumably through which these cells can migrate. So the overall model I have for ECM remodeling and interstitial cell invasion is that you have this wound that gets closed. MMP expression causes degradation of the ECM, which allows for interstitial cell migration and invasion, and then later formation of a new ECM component through collagen expression. Finally, just to return to Hydra viridissima, I wanted to show here's our normal Hydra vulgaris ECM gap, but then in viridissima, we can use that same antibody to show that we get that this ECM remodeling is conserved across the Hydra genus. So I'm really excited to follow up with that in the near future. I want to thank my lab, including some alumni whose work helped form the basis of this. Ming Zhang for providing the collagen antibody, SDB and the Hartwell Foundation for funding. Angel Perez is a really talented undergrad who has his own project, but is also helping me with some in situs as well. So thank you so much. I wouldn't go over too much.
Okay, great. Thank you, Ben, for that talk. As a reminder, if you have questions for Ben, please put those in the Q&A, and I will pose them to him as they come in. So first, I just want to say I think it's so lovely that you had the opportunity to give a little shout out to Ethel Browne Harvey right at the beginning of your talk. I didn't know about that work of hers, so that was really cool to learn about. And part of me, you made me want to go read that paper that you pulled that little quote from. So that's great. I was kind of curious about the sort of the timing of the regeneration or sort of the reestablishment of some of these different populations. Because in my work, I kind of tend to focus on like one population, and I sort of think of the other cells as just being in the environment. And I know that you're kind of working on maybe sort of a broader scale than that. But I'm just curious about how the timeline of like reestablishing these different populations in the regeneration context that you're looking at, is it at all similar to how that happens in development, or do things go out of order in this regeneration context? Like how closely does that recapitulate development?
Yes. So that's an interesting question, especially because it's kind of interesting to talk about Hydra development in general. So we don't actually know that much about Hydra embryonic development because they develop their eggs, like they have a very hard cuticle. So we don't actually use Hydra that much to study embryonic development proper. What you can study is asexual reproduction, budding, like which is just it's a different form of development. But so when so I'm actually hoping to sort of follow up this paper with a little bit of work on budding with the replenishment of cell populations in budding, because buds always come off from the bottom of the animal, and some of the cell populations that are only in the head, you wouldn't get them growing in a bud, so you would need to replenish those as well. So you would actually see some invasion. You would presumably see some invasion of those ectodermal stem cells into the endoderm. And I haven't done like thorough experiments quantifying this, but I've like looked at some sections and definitely seen what appear to be sort of like recently invaded cells, like a large population of recently invaded cells in the budding head. The overall timeline of bud formation is a little bit different. But I think broadly speaking, like they regenerate similarly insofar as like you have this head that needs to form, in order to form that, some of the nanos positive cells are going to have to invade. But yeah, we tend to see those cell populations get replenished within a couple of days. Like we start seeing markers of interstitial cell differentiation within 2 days. So some of these like the gland cell populations that are normally in the head, you don't really see them regenerated at like a day and a half, but by 2 days they're there. So that's about the time course for like seeing these cells mature.
Okay. Great. So we have a question in the Q&A here from Rob Harris. He says, beautiful images, Ben. Is it possible to increase MMP expression in certain cells or regions to break down collagen structures in the absence of amputation and show that you still find this migration of stem cells?
Yeah, so as far as increasing MMP expression in certain cells, I guess I suppose you could do that via electroporation. Like you can electroporate RNA as well as like siRNAs. So potentially you could then get you generally get like a mosaic of expression, and maybe like you could maybe alter MMP expression in patches of the animal that way. So yeah, I have not done that, but that's certainly a really interesting experiment. One of the issues is that like we have these protocols for electroporating like siRNAs or really any sort of gene modification. The issue though is that it's much harder to get them into the endoderm. Like ectodermal electroporation is pretty easy, but like you really have to blast them to get the siRNA all the way into the endoderm, and they are really not like you have to feed them a ton for like months just to get them like hardy enough to survive the amount of electricity you have to shock them with to get a siRNA into the endoderm, and that's where all my expression is happening. So gene modulation is pretty tough, but I'm hoping to get there in the near future.
Okay, great. And we have another question here from Leah. She says, great talk. Is the endoderm also secreting other components of ECM? And what is the timeline for full ECM reestablishment? Would you see similar phenotypes if you just knock down one component of the ECM?
Okay, yeah. So the endoderm is, as far as we know, it's secreting the vast majority of the ECM components. Like it does produce laminin. I mentioned before that I thought some previously published work was incorrect, and so that work had sort of suggested that it was endoderm making laminin, but ectoderm making collagen. I think that the actual answer is that endoderm is making both. There are other ECM components. So there's an interstitial lineage cells called nematocytes that have these very short collagens called mini collagens, so they make those. But in terms of the basement membrane like ECM components that comprise the mesoglia separating the two epithelial layers, to my knowledge, the all the genes that I've looked at tend to be made by the endoderm. So this seems to be really an endoderm controlled process. The only thing I could really find, even like the genes that are involved in post-translational modifications of like oxidase genes for like making the collagen. But as far as the timeline of ECM reestablishment, it tends to look pretty close to that of an uninjured animal at between 2 and 3 days. Like really, like you see this. It's very easy in most cases early on to find that little, I don't want to say scar would be the incorrect term because that has a very specific meaning in terms of collagen, but like that line where you can tell that there's a demarcation where the head is regenerating. That's pretty easy to determine where that is for the first couple of days, but afterwards it becomes harder because you filled in new collagen to the extent that there's not really a difference between the regenerating head and the uninjured portion of the animal. And then as far as similar phenotypes, I have not yet tried knocking down individual components of the ECM. As I mentioned before, the endodermal gene knockdown is a pretty difficult experiment to do, but that's certainly within my plans in the near future. Just one of those quirks of working with the Hydra that didn't know what I was getting into when I started, but I wouldn't have it any other way.
It's a great answer. Okay, I don't see any other outstanding questions here, so I think we'll wrap up. So thank you, Leah and Ben, for your excellent talks. This seminar has been recorded and it will be available on the SDB website next week. Please join us for next month's seminar, which will happen on Friday, November 8th. When Yuchi David Chen from New York University and Carrie Weber from the University of California, San Diego will be presenting. And thank you all for coming.